Hi everyone, today we're going to be talking about Mendel's laws by That High School Tutor. To understand where Mendel was coming from, we have to go even back. So, we have the first few theories that were about genetics. So, starting in 400 BC, we had pangenesis, where Hippocrates thought parts of an organism go to their gamete, such as their skin color, go inside their sperm or egg, etc. And then the other theory was blending, wherever materials from an organism literally blend to create another offspring. Now, there is an inheritance that is somewhat like this, but not in Mendelian. Now, let's talk about Mendel. He came along and he said that parents pass heritable elements. Now, what are these elements called? Genes. So he chose pea plants to observe. Why, you might ask? Because each characteristic happened in two contrasting traits. I have a chart here, for example. So they're very different from each other. We have a flower color, one is purple, one is white. A seed color, one is yellow, one is green, one is round, one is wrinkled. It's one or the other. There's no in between. So to understand how Mendel created his experiments, we have to know the anatomy of the flowers. So the flowers actually have both the male and female reproductive systems in it. The male reproductive system is called the stamen, and then the female reproductive system is called the carpel/pistil. This is why these flowers usually self-fertilize. Now, in self-fertilization, there isn't a lot of diversity. So, that's why Mendel used a different method. Here's a diagram that goes into very detail on each part of the flower. We have the pistil and we have the stamen. This is also important that pollen is the sperm of the flower, and the egg are in the ovaries, like female humans have. So, what did Mendel use as his method? He used cross-fertilization, which basically means that he transferred pollen between two different flowers to create that diversity. So this is a diagram on how he did it. So we first removed the stamens from a flower, in this case purple, and he transferred pollen from the stamen, which is, remember, the male reproductive system, of the white flower to the carpel, which is the female reproductive system of the purple flower. As you can see, a paintbrush here taking the pollen, taking it all the way to the carpel. So he pollinated the carpel, and this carpel matured into pods. And then you see the diversity here. So, his experiments. He bred plants for many generations that were true breeding, or called purebred, for specific traits. This is called the P generation. Now, that was a mouthful. So it's basically saying that they bred the same type of plant. So, like, if the offspring was a tall plant, the only way they got that tall plant is by breeding two tall plants. So if you need to, this is the first step in the experiments, the first generation. If you need a sort of reminder, or if you need some sort of, like, I'm not sure what to call it, thing, to remember that the P generation is the first generation, just think of P equals parent. Since the P generation is also called sometimes the parent generation. So, after this, he crossbred the P generation and then called that offspring the F1 generation. But in the next offspring, he did something different. He let the F1 generation self-fertilize, which is the key point, and called this offspring the F2 generation. This quite literally worked by luck, and it worked on his first try. If he chose some other plant, I'm not sure, it might not have had as successful, and might not have been as successful as him choosing the pea plants. This was literally done by pure luck. Of course, there was some science, but without choosing the right plant, So, here's a pic that somewhat explains, and I can walk you through. So, this is called the parents, or ding ding ding, the pea generation. As we see here, we have a tall plant and we have a short plant. Now, this tall plant is probably the offspring of two other tall plants, and this short plant is the offspring of two short plants. So we see that the offspring of these two from cross-pollination or cross-fertilization is two tall plants. We call this generation the F1 generation. Now, from these two plants, they produced 4 offsprings through self-fertilization. Not cross-pollination, but self-fertilization. And you see here that the short plant shows up again. Even though it wasn't showed up in the F1 generation, it showed up in the F2 generation. To learn on how this is possible, we use Punnett squares. If you want to know more about Punnett squares, I'll leave a link in the comments on how we use them to distinguish how we get these traits. So this was his explanation on why this was happening. An organism has 2 copies of an inherited gene, one from each parent, which makes sense because in our gametes, we make 2 copies. Another form of genes are called alleles, and they are 2 different types of alleles, as we've seen in the 2 different traits. We have the dominant alleles, and we have the recessive alleles. Dominant alleles are expressed over recessive alleles, which you can tell by the names. We represent dominant alleles by a capital letter. It does not have to be D, but it has to be the same letter. And recessive alleles are represented by the same letter, but lowercase. Now, he came up with 2 ways, or as most famously known, Mendel's laws. He came up with the law of segregation and the law of independent assortment. The law of segregation states that only one of the 2 gene copies present in an organism is distributed to each gamete. That makes the distribution of the copies different. And the law of independent assortment states that the alleles of 2 different genes get sorted into separate gametes. Now, that was a mouthful, and I like to use a picture to really understand what I'm talking about. So here's a really good picture that shows the difference. Now, to mention beforehand, genes can be the same. Like homologous chromosomes can have the same genes, but they can have different alleles. So that's why we represent these alleles with different letters, dominant being capital, recessive being lowercase. Anyway, in the law of segregation, the 2 alleles from the gametes here, or the germ cell, separate to produce these 4 gametes. But we see here that even though the homologous chromosomes have lined up, the red have gone with the red into 2 of the gametes, and the blue have gone with the blue into another 2 of the gametes. So this shows that they have somewhat of the same traits in each of these and the same traits in these 2. But we see here the same thing, that we have Y, capital Y, lowercase r, capital Y, lowercase r, which are the different alleles. But in this case, we have the alleles of genes on non-homologous chromosomes. and they assort independently during their formation of gametes. So we see red and red are actually flip-flopped, and we have the R and the Y flip-flopped from this. That's why it serves as different genetic variation. We see here, instead of it being two capitals, we have one capital and one lowercase. These were these two laws on how genetics had worked. If you want to learn more about Mendel's laws in more depth and more explanation, I have some links below to help you on that. And that's it for Mendel. Thank you so much for listening, and if you have any questions, just put them in the comments. Bye!